Brief introduction on enzymes. First, enzymes are a type of protein used to catalyze or speed up reactions. They do this by lowering the activation energy required to start a reaction, and they also help regulate reaction rates. So in the duodenum, trypsin catalyzes the hydrolysis of peptide bonds and breaks down proteins into smaller peptides. The peptide products are then further hydrolyzed into amino acids via other proteases, rendering them available for absorption into the bloodstream. Trypsin is a proteolytic enzyme important for the digestion of proteins. Trypsinogen is made in the pancreas and then enters the small intestine via the common bile duct, where it's converted into active trypsin. The pH, where it's normally functioning in the body, is around 7.4, and the temperature would be around 40 degrees Celsius. So the question we were asking was, does temperature change affect trypsin's ability to perform? We believe that it will affect the enzymes, and the temperature other than the normal body temperature, trypsin will not be able to perform normal. Our hypothesis was that we believe trypsin does not affect enzyme activity, or it does affect enzyme activity, as the temperature gets higher and lower than 37 degrees Celsius, the enzyme will not begin to work properly. So we believe this because once you change conditions trypsin normally functions in, it will not be able to adapt to the change, and therefore it will cause it to denature. We also could have tested pH and salt concentration. So here are the materials we used. First is the reaction buffer, the azacasine. We used trypsin, and for our controls, we replaced that with distilled water. We also used the heat block to get our different temperature intervals, which were 47, 52, 57, 62, 67, and 72 degrees Celsius. We used the centrifuge and the spectrophotometer to record wavelength. So our methods were, first we added reaction buffer, azacasine, and trypsin to each test tube. The azacasine was the substrate used, and the sulfanilamide groups are added and covalently bond to the peptide bonds found in the milk proteins in the azacasine. These groups affect the color of the supernatant, so the darker the orange, the more free-floating bonds, and the lighter the less. Next, we incubated for 10 minutes, and then we added TCA, which was used to stop the enzymes from precipitating. Next, we centrifuged the test tubes. This was done after adding TCA to remove undigested substrate and what was in the pellet and supernatant. Next, we transferred the supernatant to a clean cuvette, and like I said before, the darker the orange the supernatant, the more free-floating peptide bonds or chains and amino acids suspended in the solution. Once that was done, we made sure that the spectrophotometer was blanked, and we recorded the wavelength for each test tube of the absorbance. So the experimental variable we were testing was the change in temperature and how it affects trypsin's activity. Our controls were pH, which we tried to remain around 7 for neutral, and the amount of reaction buffer we used. Our negative controls, we added trypsin to one set of experiments, and for the control group, we replaced the trypsin with distilled water just to see if the trypsin had any effect or if there was any possible other things affecting the activity. The reason for replicating, like I was saying before, was to ensure that the breakdown of the azacasine wasn't caused by the trypsin, but was only due to the temperature change, which was later confirmed in our data. So the major changes we did from the first time testing this was using replicates so we could compare the results and ensure that they were accurate. We also changed the temperatures we used, and we used a much wider range of temperatures to be tested. So here are our results shown. By using the t-test, our results show that every temperature had a significant difference except between 47 and 52 degrees Celsius. This can be shown in the graph where the standard deviation lines overlap. There was an error in our data where the absorbance went up at 72 degrees, but after we compared our results with other groups, we found that they also experienced the same error, so this could have possibly been due to an error with the heat block. So basically, like everything I was stating before, we were testing temperature's effect on trypsin. And the results showed that trypsin can work in higher temperatures than normally found in the human body. So our discussion, our hypothesis was not supported by our results. We believed that trypsin couldn't work outside of normal body temperatures, which would be 37 degrees initially, but from our results, we found that it can actually work of temperatures up to around 52 degrees. Our data showed that as the temperature increased, the trypsin's function was altered, and thus supporting trypsin's optimal temperature is actually higher than what we may have tested. Referring to our t-test results, our data showed that with how close the deviations are between 47 and 52 degrees, the optimal temperature may still be unknown. So looking forward, we could have possibly tested smaller intervals instead of going and increasing by 9 degrees Celsius for each one tested. This is shown in the graph where the standard deviation lines overlap in the graph previous. According to our graph, it is possible the optimal temperature could be somewhere around 57 degrees Celsius due to the decline in absorbance at higher temperatures. In relation to the pH we were using and our choice of control variable conditions, we feel that our experiment did not result in the highest possible rate of enzymatic activity. In our experiment, the exact optimal temperature was not actually found. Only an assumption was made due to the insignificant deviations between the 47, 52, and 57 degree marks. In the group that tested pH, their experiment consisted of variables on scales from a pH of 5 to 10, and we felt this more accurately found the highest rate of enzymatic activity because there was no gap in between the numeric values of their data set like there was when testing temperature. The intervals were just so large. And this is our t-table with averages and standard deviations shown. Yeah, these are just the t-test value results.